Hey everybody, welcome back to another video. Now, I've actually been a little bit inactive on YouTube, and that's because, as I mentioned in my previous video, the trials are starting and there's a lot to do, a lot of testing, a lot of stuff related to shipping, etc., etc. So that's why there haven't been any new videos recently. Before I begin, I would like to thank all of my YouTube members. Thanks to you, I'm able to spend the majority of my time working on the Susan Shore device project and on my research. And so just huge thanks to everybody who is supporting me. Now, today we're going to be talking about the variability in spike timing in the context of spike timing dependent plasticity. Now, I'll try to explain this as easy as I possibly can. So my recommendation is everybody watch all of my previous videos to understand what I'm talking about in this one. Now, a brief overview. What is spike timing dependent plasticity? Well, it is a form of synaptic learning rule where the precise millisecond timing between pre- and post-synaptic spikes determines whether the synapse is strengthened, long-term potentiation, or weakened, long-term depression, LTP or LTD. In classical Hebbian STDP, spike timing dependent plasticity, if a presynaptic neuron fires a few milliseconds before a postsynaptic neuron, the synapse is potentiated, often requiring a pre to post interval on the order of maybe 5 to 20 milliseconds, whereas if the presynaptic spike arrives after the postsynaptic spike, the synapse undergoes depression. If you imagine two neurons right next to each other, the presynaptic neuron is the one that occurs before the postsynaptic neuron in a traditional neural pathway or directional pathway. STDP operates within a narrow temporal window of tens of milliseconds. And this reflects the kinetics of synaptic currents and intracellular signals, like the very important calcium-dependent signaling pathways, that register the coincident activity. And this mechanism provides a very significant computational advantage. It allows for neural circuits to learn temporal sequences, to refine their spatiotemporal response properties, and encode causality of events, for example, in the external world. So it's very useful overall for our brain's properties, especially learning properties. Now, for example, when developing sensory cortices in developing organisms, STDP contributes to the formation of orientation selectivity or tonotopic maps by strengthening synapses that consistently fire with the right timing during sensory events, while weakening those with poorly timed activity. And this timing-dependent refinement, or these refinements, they underlie associative memory formation and perceptual learning in neural circuits. So you can imagine it as, for example, a person who is learning something, and he is repeating the same thing over and over again. He is associating this memory with different things, like, for example, when a person is learning new words, you can associate this word with something else. And I use this always when learning something new in neuroscience. For example, let's take a very classical look, the hippocampus in the brain. What does a hippocampus sound like? It sounds like hippopotamus. Yes, so we can, for example, associate this term with the image of a hippopotamus, and we can, of course, repeat this over and over again. And some people even like having little rhymes and little poems to remember these terms even better. And of course, the constant strengthening of synapses associated with this certain thought or this certain term that includes not only association but also repetition will lead, of course, to this memory kind of staying in the brain, so to speak, longer and more reliably. Now, in neuroscience, a lot of the principles of the brain's function are based on the fact that the brain is very energy efficient. So why waste energy on something that is unimportant when it can focus on something that is more important? And this not only is related to memory and thought, but also to other things like movement or any somatosensory actions. Now, while spike timing-dependent plasticity has been experimentally observed in many different types of neurons and synapses, it is not universally applicable to all neurons or synapse types. Its applicability and mechanisms depend on the specific properties of the neural circuits and synapses in question. So, for example, different neurons exhibit distinct STDP rules depending on their intrinsic excitability, connectivity, and, of course, functional roles in neural circuits. Of course, there's also the difference in synapses, like, for example, excitatory versus inhibitory, or chemical versus electrical, may also exhibit different forms of plasticity, some of which do not exactly 100% strictly follow the STDP rules. And of course, most importantly, STDP relies on molecular mechanisms like NMDA receptor activation, calcium dynamics, or those calcium-dependent signaling pathways I mentioned, and of course the protein signaling pathways as well. And if a synapse lacks the necessary molecular components, then STDP may not be possible. But in the context of tinnitus, we know, thanks to Susan Shore's research, that plasticity is exhibited in the SP5 to DCN synapses, or the parallel fibers, which are long extended axons that come from the granule cells from the somatosensory pathway, and the apical dendrites of the fusiform and cartwheel cells of the dorsal cochlear nucleus. Now, in the context of cartwheel cells, fusiform cells, and the parallel fibers of granule cells, if we're talking about in vitro experimentation, which basically means putting these neurons in a test tube and testing them, so to speak, locally, we can actually understand that the calcium-dependent signaling pathways required either for LTP or LTD induction are absolute. So if you guys take a look at my linear versus Susan Shore device video, one of my very first kind of major scientific videos, I explained there that for long-term synaptic depression to occur, very specific timing rules at the synapse must be followed. In this case, it would be the postsynaptic spike occurring very, very briefly before the presynaptic spike. So maybe 5 to 10 milliseconds. Or for LTP to occur, it would be the opposite. The presynaptic spike occurring briefly before the postsynaptic spike in rapid succession. And like I mentioned in my previous video, there's different types of enzymes that are activated during these specific timing rules. For example, for LTD timing rules, or anti-Hebbian timing, which means the postsynaptic spike occurs before the presynaptic spike, phosphatases are activated. And there's different types of phosphatases, like protein phosphatase 1, PP1, protein phosphatase 2A, PP2A, or calcineurin. And what these actually do is they remove phosphate groups from proteins, lipids, and other molecules. This is a process known as dephosphorylation. And dephosphorylation of I guess you could say, anchoring proteins of the AMPA receptors on the cell membrane means that these AMPA receptors will be removed and internalized into the nerve cell, which means that this specific neuron will become less responsive to any presynaptic spike. And in the context of tinnitus, this would mean that the level of synchronization or synchronized firing of the neurons will decrease, which means that the thalamic gating mechanisms in the thalamus would adequately filter this aberrant noise, or not recognize it as a significant auditory event, not passing it further up to the auditory cortex. Now, if we're talking about long-term potentiation, which we actually do not want, That would be the presynaptic spike occurring briefly, or in a brief time window, before the postsynaptic spike in rapid succession. This activates CAMK2, or calcium/calmodal-independent protein kinase 2. Now, kinases are, of course, enzymes that do the opposite of phosphatases. They phosphorylate proteins by adding phosphate groups to them, which means that the protein anchors of the AMPA receptors on the nerve cell membrane become stronger, meaning the AMPA receptors will not leave the cell membrane as easily, and potentially even more of these AMPA receptors will be added to the cell membrane due to this, which means that the responsiveness of this specific nerve cell will be increased, which would mean, of course, more hypersynchronization, which would be the complete opposite of what we want and what I explained earlier with LTD induction. Now, as I mentioned before, in vitro or in a test tube, these mechanisms are absolute, or at a synaptic level, they are absolute. So for these different enzymes to be released, the postsynaptic and presynaptic timing must, of course, be absolute. But if we're talking about in vivo, or in living organisms overall, there are many different things that can potentially affect the spike timing, or the spikes in general, of the neurons. This includes, of course, variability in excitatory and inhibitory balance, which is something that people with tinnitus or other conditions have severe dysfunction in. This can also be based on different variability in ion channel function, like HCN channels, or, for example, potassium channels, etc., etc. And all of these things actually change the spike timings of these neurons. But more on that later. Let's actually talk right now about in vivo examples of the variability in timing rules. Now, one thing that people do not really think about is the afferent conduction delay and its impact on spike timing in vivo. So what is afferent conduction delay? Afferent conduction delay refers to the transit time for action potentials to travel from a presynaptic neuron's soma to the synaptic terminals of a postsynaptic cell. And this delay is influenced, of course, by multiple factors, one of which is the axon length and diameter. Now, longer pathways naturally, of course, introduce more delay. One example of a pathway is, of course, from the cochlea to the dorsal cochlear nucleus, or from the trigeminal nerve to the dorsal cochlear nucleus. Now, in general, longer pathways, as I mentioned before, they introduce more delay. And thin or unmyelinated axons, they conduct impulses slower than thick myelinated ones. For example, unmyelinated fibers conduct at maybe 0.5 to 10 meters a second, whereas large myelinated axons can reach velocities of up to 150 meters a second. So an excitatory input coming from a distant, slow-conducting axon might arrive tens of milliseconds later than one from a fast and local fiber. And this difference can be very significant relative to the very critical STDP window, which is no more than maybe 10, 20 milliseconds, and in our case, even less, 5 to 10 milliseconds, meaning conduction velocity disparities can actually flip the effective spike timing order. So a synapse with a longer delay might turn what would have been a pre-before-post pairing into an apparent post-before or apparent post-before pre-pairing. So LTD or LTP from the postsynaptic neuron's perspective. And if any other input triggers the postsynaptic spike sooner, and small variations in conduction due to many factors can shift the timing between pre- and postsynaptic spikes on a synaptic level, and it can actually tip the balance between potentiation and depression at the synapse. And just like conduction in electricity, conduction in neural circuits can also actually depend on the external environment. So, for example, whether or not the environment is cold or the environment is warm. There, of course, being more delay if the person is in a very cold environment compared to a warmer one. So, a simple example, let's say somebody has very severe damage to their cochlea. This, of course, would increase the conduction delay, whether that be by a couple of milliseconds or maybe even tens of milliseconds. And if a person is using a bimodal stimulation device, this would mean that their auditory pathway is, of course, damaged, which means that there is more conduction delay, meaning that the potential postsynaptic spike could arrive later than necessary, or later than the default timing, of course. And as we know on a synaptic level, as I explained before, the spikes must arrive at the precisely correct time. And if there is a delay due to neuronal damage, which we will talk about in a second, this would, of course, disrupt the very strict timing rules. And in the context of myelination and nodal architecture, the degree of myelination and internode spacing affect conduction velocity, and demyelination, like in disease or nerve injury, slows conductance and can increase variability or jitter in spike arrival times. And in an STDP context, demyelinated afferents might consistently miss the narrow LTP window, falling into the LTD window instead. And on the other hand, highly myelinated fibers fire with tight timing precision, potentially reinforcing consistent STDP outcomes. Meaning in some very specific context, an external Hebbian timing, or timing that would supposedly introduce LTP, in a damaged auditory pathway, or a severely damaged one, could technically lead to LTD based on this variability in afferent conduction delay. Now, if we talk about synaptic integration and dendritic delay, even after an action potential reaches the synaptic terminal, there is still a synaptic delay on the order of maybe 1 to 2 milliseconds for neurotransmitter release, and then a postsynaptic membrane integration time. And postsynaptic potentials, or PSPs, have finite rise and decay kinetics. In other words, an AMPA receptor-mediated EPSP might rise within a few milliseconds and decay over tens of milliseconds. And if the synapse is located on distal dendrites, additional delay occurs as the EPSP spreads to the soma. Now, neurons with long membrane time constants or slow dendritic propagation, they effectively blur the timing of synaptic events. If a postsynaptic neuron integrates inputs slowly, a presynaptic spike that arrives slightly earlier might still be contributing to depolarization by the time the neuron fires, extending the effective timing window for STDP. On the other hand, fast integration or strong feed-forward inhibition, shaping the EPSP, can narrow the window. Therefore, variability in dendritic processing time due to the morphology or receptor kinetics will also alter the relative pre-post spike timing that the synapse experiences. So basically, variability in afferent conduction can dramatically influence whether a given spike pairing falls into the depression or potentiation window. Computational models and experiments do show that STDP can select for specific axonal delays. In other words, connections whose conduction times naturally line with postsynaptic spiking get potentiated, while those out of phase get weakened. For example, precise axonal delay lines in the brainstem can be tuned for sound localization, and STDP modeling predicts that only synapses with the correct conduction delay or matching the intraoral timing difference are potentiated, which helps the circuit lock in on a specific time difference or disparity. Generally speaking, networks under STDP can potentially self-reorganize in such a way that appropriate conduction delays that reliably cause presynaptic before postsynaptic firing are reinforced. In a context of tinnitus, that would mean the tinnitus spiking. Basically, overall, what does that mean for Susan Shore's device? In essence, a single STDP time rule is generally insufficient. Due to all of these reasons, like conduction delays and physiological context bias, the induction of LTP or LTD, meaning that adjustments to something like, for example, the bimodal interval would potentially be necessary for the treatment. Bimodal interval being the delay between the, for example, auditory and somatosensory stimulation. Now, for any of you heroes who are still watching this video, let's talk about neurotransmitter dynamics and their role, of course, in LTD induction. Of course, induction of timing-dependent LTD relies not only on the spike timing and the afferent delays, but also on the postsynaptic biochemical cascade that is triggered by the neurotransmitters. The primary excitatory neurotransmitter, glutamate, has a dual receptor action that is really important in STDP. Now, AMPA receptors mediate fast EPSPs, the excitatory postsynaptic potentials that depolarize the postsynaptic neuron within a millisecond of glutamate release. And this rapid depolarization can contribute to bringing the neuron to a threshold or removing the magnesium block from the NMDA receptors, which will lead to a full-blown action potential or spike being fired. However, AMPA currents are brief, and if a presynaptic spike arrives after a postsynaptic spike, by the time glutamate is released, the postsynaptic membrane may have just repolarized from the spike or even be hyperpolarized due to after-spike potassium currents, meaning the AMPA EPSP will be less effective. And in this scenario, postsynaptic spike that precedes the glutamate release tends to favor LTD. In other words, the glutamate still binds to AMPA or NMDA receptors, but the postsynaptic cell, no longer strongly depolarized, allows NMDA receptor activation only weakly. NMDA receptors, of course, are the critical coincidence detectors in STDP. They require both glutamate binding and depolarization to relieve the magnesium block. And when they do open, NMDA receptors admit the calcium ions into the dendritic spine, which triggers the intracellular signaling that decides LTP versus LTD. The kinetics and subunit composition of NMDA receptors can, thus, sway the timing requirements for plasticity. Now, if we talk a bit more about NMDA receptor dynamics, different NMDA receptor subunits, like NR2A versus NR2B, for example, they alter the channel's open duration and calcium influx profile. NR2B containing NMDAs generally have longer open times or slower decay and allow more calcium per activation than NR2A containing receptors. And during development, for example, many cortical synapses shift from predominantly NR2B to more NR2A, raising the threshold for plasticity induction. And a key idea is that the ratio of NR2A versus NR2B tunes the LTP or LTD threshold. It doesn't necessarily dictate the direction of plasticity overall or outright, but it controls how easily a given pattern triggers potentiation or depression. So basically, if NR2B is high or low NR2A slash NR2B ratio, even modestly correlated pre-post activity can cause large prolonged calcium transients, which favor LTP and effectively lower the threshold for potentiation. And in such cases, the window for LTD, which requires smaller calcium signals, is reduced because almost any coincident activity produces large or a massive calcium influx that activates CAMK2 or the kinase for LTP, which basically means that a person who has more reactive tinnitus or hyperacusis, for example, is more prone to LTP. And on the other hand, if NR2A is high, NMDA receptor currents are shorter or less calcium per spike. So a pre-post pairing might only produce a moderate calcium elevation, so insufficient for LTP, but enough to activate phosphatases, yielding LTD. Synapses with more NR2A require tighter timings or higher frequency firing to achieve LTP and are biased towards depression for casual spike pairings. And this molecular tuning means that the timing requirements for LTD induction are not fixed, but depend on receptor properties. In other words, a slow NMDA receptor or NR2B rich might still summate calcium from a pre-post interval, whereas a fast NMDA receptor or NR2A rich one might only summate within approximately, for example, maybe 20 milliseconds. Now, generally, modeling work does suggest that to reproduce LTD, NMDA channels must have relatively slow kinetics and specific calcium feedback regulation. So if NMDA currents shut too quickly or don't get properly modulated by calmodulin, LTD may fail to induce. Now, about GABAergic modulation. Now, inhibitory neurotransmission, mostly through GABA, powerfully controls spike timing and, of course, STDP outcomes. GABA-A receptor-mediated currents can hyperpolarize or shunt the postsynaptic neuron, basically affecting how precisely spikes occur relative to EPSPs. For example, feed-forward inhibition that arrives immediately after an excitatory input can truncate the EPSP and prevent or delay postsynaptic firing, basically narrowing the time window for LTP, for example. And more interestingly, some experiments show that GABAergic circuits can even invert the polarity of STDP by altering the excitability timing. And in the striatum, blocking GABA-A receptors was found to reverse the usual STDP timing rule. Now, normally pre to post firing yields LTP and post to pre yields LTD, but when GABA is blocked, the opposite timing produces plasticity. And this means that in the intact circuit, GABAergic intraneurons timing the inhibition are effectively offsetting the spike timing requirements. And how is that? One mechanism is through the after effects of inhibition on the dendrites. And, for example, distal GABAergic inputs can cause a rebound depolarization or delay the repolarization of dendritic membrane, thereby shifting the phase at which an EPSP will induce a spike. Now, if GABA timing is altered, like by disinhibition in a disease state, the postsynaptic neuron might fire earlier or later than usual in response to an EPSP, which could convert what was an LTB pairing into an LTD pairing, or vice versa. Basically, GABA controls STDP timing precision and polarity by regulating neuronal excitability between spikes, pretty much. And in cortical networks, for example, the balance of excitation and inhibition sets an operating regime. If inhibition is strong and precisely timed, only tightly synchronous presynaptic spikes can make the neuron fire, favoring a narrow STDP window. If inhibition is reduced or mistimed, the neuron might fire more freely, even to slightly out of sync inputs, effectively broadening the window or altering which spike order dominates. So basically, changes in GABA release or receptor function, like those observed in aging or tinnitus models, where inhibition is downregulated, can make timing-dependent LTD either easier or harder to induce by changing the background excitability of neurons. Now let's talk about cholinergic and serotonergic influences. Now, neuromodulators like acetylcholine, or ACH, and serotonin, or 5-HT, do not carry specific sensory information, but they do modulate the electrical properties of neurons and the biochemical gating of plasticity. Now, these modulatory transmitters can gate whether STDP occurs at all and adjust the required timing or repetition for LTD or LTP. For example, in the visual cortex, the presence of acetylcholine, which can be stimulated with a cholinergic agonist like nicotine, has been shown to convert a timing rule. An STDP protocol that would normally produce LTP instead produced LTD when nicotine ACH receptors were activated, and specifically in layer 2/3 or 5 synapses of the prefrontal cortex, nicotine application flipped the timing of LTP to LTD. Now, this is thought to happen because HCH can activate muscarinic receptors that affect potassium currents, broadening postsynaptic spikes or depolarizing the resting potential, and also because ACH receptors on presynaptic terminals can change release probability. And the net effect is a change in the integration window or in the relative weight of pre versus post firing needed to trigger plasticity. Now, serotonin similarly can modulate membrane conductances and intracellular cascades. For example, via 5-HT2A receptors, it can depolarize neurons, or via 5-HT1A hyperpolarize them, thus either enhancing or reducing excitability. And neuromodulators often engage second messenger systems that alter the phosphorylation state of ion channels and synaptic proteins. And as a result, they can broaden or narrow the STDP window, again, and raise or lower thresholds. Now, another clear example is activating beta-adrenergic receptors, or norepinephrine, in the hippocampus, broadened the LTP window from 10 milliseconds to 15 milliseconds without changing the maximum LTP magnitude, implying that norepinephrine made the synapse sensitive to more timing jitter, likely by increasing excitability or augmenting NMDA currents. In general, neuromodulators like ACH, norepinephrine, dopamine, serotonin are known to act as permissive gates for plasticity, often required for STDP to be included in the adult cortex, for example. No neuromodulator equals the synapse might be immune to plastic change, which is very important. Keep this in mind. And they can control the polarity, as seen with nicotine, and the temporal requirements for STDP. The underlying mechanisms involve neuromodulator-induced changes in calcium dynamics within the neuron. For instance, ACH can activate muscarinic receptors that close the potassium channels, leading to broader spikes and more calcium entry per spike. And dopamine or norepinephrine can modulate NMDA receptor phosphorylation, or internal stores of calcium ions, or IP3-mediated calcium release, by ultimately influencing postsynaptic calcium transients. Neuromodulators dictate whether a given pre- or postsynaptic timing results in the high calcium spike needed for LTP or the moderate sustained calcium spike that favors LTD. Therefore, in the context of LTD induction, high cholinergic or noradrenergic tone might make it harder to induce LTD because they push towards bigger calcium signals and LTP, whereas the absence of neuromodulators or presence of others might bias towards depression. And this is highly pathway and receptor specific. But the take-home message is that STDP is not static. Once again, the timing rules for LTD and LTP can shift dramatically depending on the neurotransmitter environment. And for precise spike timing dependent LTD to occur, often the neuromodulator tone is low. No expectation or attention, whereas learning a task, for example, high attention or high ACH or norepinephrine, may tilt plasticity towards potentiation or require pairing with reinforcement signals. And basically, understanding these dynamics is very important because it suggests that we can pharmacologically, for example, bias synapses towards LTD by manipulating neurotransmitter systems. For example, blocking cholinergic inputs during therapy might widen the time window for inducing LTD on hyperactive synapses in, for example, tinnitus or epilepsy. Now let's talk about mechanical considerations. Now, STDP experiments and therapeutic stimulation can involve either natural stimulation, in other words, sensory input like sound vibrations to the cochlea, or artificial electrical stimulation like with electrodes. And these two modes can recruit the neural circuits in very different ways, leading to variability in spike timing and plasticity outcomes. Now, if we talk about natural mechanical stimulation in auditory circuits, for example, the cochlea, imagine how sound is normally transduced. The acoustic vibrations move the cochlear baseline membrane, and the hair cells convert this mechanical deflection into receptor potentials and neurotransmitter release. And the auditory nerve fibers fire spikes phase-locked to the sound for lower frequencies, for example. Just watch my video on how fusiform cells process sound at different frequencies. And this process imposes specific delays. For example, high-frequency sounds cause base hair cells to respond almost immediately, or less than 1 millisecond of synaptic delay, whereas lower frequencies or distant hair cells might add a slight delay. Moreover, sound intensity and the cochlear amplifier, or the outer hair cells, can modulate the timing and synchrony of auditory nerve firing. And typically, not all auditory nerve fibers fire at once. Their spikes are distributed in time according to the sound waveform and their characteristic frequency. This natural jitter means that postsynaptic targets, like the cochlear nucleus neurons, receive a slightly distributed barrage of inputs rather than a simultaneous volley if we're talking about broadband noise. And the timing between excitation of different neural pathways is thus determined by the physiology. For example, a loud click might synchronously activate many fibers with minimal delay differences, whereas a complex sound like broadband noise yields a more temporal spread. And this is actually really important in the context of the Susan Shore device, and why stimulating, for example, all the frequencies at once is not a good idea due to this variability. Now, additionally, natural stimulation often co-activates modulatory feedback loops or afferent fibers to the cochlea or multisensory inputs, of course, that can influence the postsynaptic neuron's state, like with usage of the Susan Shore device. Now, if we talk about the electrical stimulation, for example, in the context of stimulating the auditory nerve fiber directly with an electrode, this bypasses the receptor and directly depolarizes the auditory nerve fibers. And this can produce non-physiological synchrony and recruitment patterns. For example, an electrical pulse delivered to the auditory nerve can activate a large population of nerve fibers near simultaneously, especially biased to the largest, most excitable fibers, which in natural hearing might correspond to high threshold or specific characteristic frequency fibers. This can create an extremely synchronous presynaptic spike, very similar to a broadband noise, by the way, arrival at the cochlear nucleus, which is far more synchronous than most natural sounds would cause. And from an STDP perspective, such massive synchrony or such massive synchronous input, like with electrical stimulation or, for example, with broadband noise, might make it easier to induce LTP in one shot, since a strong synchronous EPSP can cause immediate postsynaptic spiking. On the other hand, if one is attempting to induce LTD via carefully timed pre-post sequences, electrical stimulation must be very precisely controlled. So does the frequency width, for example. Otherwise, the synchrony might drive the postsynaptic cell to fire too early or too robustly. And if we talk about electrode placement, this matters greatly as well. Stimulating the afferent fiber bundle versus stimulating near the postsynaptic cell can result in different latency. For example, in an auditory brainstem slice, for example, placing the electrode on the auditory nerve root will introduce the natural axonal conduction delay to the cochlear nucleus, whereas placing the electrode right in the cochlear nucleus will directly excite the cells with essentially zero axonal delay. Now, of course, we are not in vitro, we are people, so if the goal is to mimic before post pairing, the person must account for where the spike is initiated, and this means on, for example, which part of the face, which branch of the trigeminal nerve, for example, placing the electrode farther from the trigeminal nucleus, for example, could induce more synaptic delay. Even if it is a variation of 1 to 2 milliseconds, this can also be very, very important for initiating the mechanisms required for spike timing dependent plasticity to occur. And, if you, for example, place the electrode way too far and you're also like in a cold environment and just, something like that, this massive delay might actually cause potentially the presynaptic spike to occur after the postsynaptic spike, even if the stimulation is incorrect or simultaneous, due to this conduction delay. So this is also very important. So let's say a person is sitting in a freezing environment. Let's say he has also a bunch of damage to his neurons or whatever. And he uses the default signal timing for Susan Shore. Now, even the Susan Shore device, even though the auditory stimulation occurs first, the window between the stimulation is very small. And if their afferent conductive delay is too high, either in the somatosensory or auditory pathway, this means that the postsynaptic spike could occur too late in the LTD window, or for example, the auditory or the fusiform cell, cartwheel cell spike would occur also too late. So that means that either LTP could be induced or nothing could happen because the LTD timing window is not hit. So for all those people wondering if electrode placement matters, it absolutely does. And if all people are also wondering, why can't we just play like a broadband noise from 100 hertz to 8 kilohertz or something, that is also the explanation why this is not a good idea to do. Now let's talk a little bit more about calcium signaling pathways in LTD induction. Now, once pre- and postsynaptic spikes occur within a given timing, it's the postsynaptic calcium dynamics that ultimately dictate whether LTD is initiated. Now, LTD, especially NMDA receptor-dependent LTD, common in, for example, the hippocampus, the cortex, or even, for example, in the brainstem as well, generally requires a moderate prolonged calcium elevation in the postsynaptic dendrite spine. In contrast to the large brief calcium burst that triggers LTP, the NMDA receptor is primarily the source of calcium for timing-dependent LTD or LTP. So the interplay of spike timing, which controls NMDA activation and receptor properties, leads to a certain calcium profile. If that calcium stays below the threshold for kinase activation but above the baseline, it preferentially activates protein phosphatases, like I said, for LTD. And key calcium-dependent phosphatases include calcineurin, also known as PP2B, PP1, and PP2A. And these enzymes, when activated by calcium or calcimodulin, remove the phosphate groups from the target proteins in the postsynaptic density, leading to the synaptic weakening, for example, by dephosphorylating AMPA receptors or their anchoring proteins, which can cause the AMPA receptors to be removed from the synapse, like I said near the beginning of the video. Now, empirically, inhibitors of PP1 and PP2A block LTD induction, showing both are required for LTD expression in CA1 synapses. Moreover, blocking these phosphatases prevents the characteristic dephosphorylation of CREB, a transcription factor substrate that accompanies LTD. So this underlines that a cascade of dephosphorylation events underlie LTD. Now, in contrast, LTP is accompanied by increased phosphorylation of many of the same, actually, substrates via CAMK2 and other kinases. In fact, a classic description is kinase activation to LTP, phosphatase activation to LTD. Now, how does timing play into this? If a presynaptic spike arrives slightly after the postsynaptic spike within tens of milliseconds, the postsynaptic cell was recently depolarized but is now on the downslope of its spike after potential. The NMDA receptors will open. Glutamate is present from the presynaptic spike, but the postsynaptic voltage is not very high. It might even be hyperpolarized due to potassium channels opening after the spike. Now, this means that calcium entry through NMDA receptors is limited enough to activate calcineurin, which has a high affinity for calcium or CAM, and can be activated by lower calcium levels, but not enough to strongly activate CAMK2, which requires higher calcium transients and stays active when autophosphorylated by large calcium events. Now, calcineurin, once active, will dephosphorylate the key sites and also activate PP1. Calcineurin can trigger a cascade that inhibits an inhibitor of PP1, thus disinhibiting PP1. This leads to AMPA receptor dephosphorylation and internalization, which is the hallmark of LTD, like we discussed in my first video. Now, one hypothesis is that the crossover point between LTD and LTP induction is governed by whether calcineurin gets activated without sufficient CAMK2 activation. A low to moderate calcium rise, for example, due to a small number of NMDA channels open over a longer time, will favor the phosphatase route. LTD is induced when calcineurin, or PP2B, is activated while CAMK2 is not maximally turned on. This is very important. And on the other hand, a large rapid calcium influx, as with presynaptic spike just before the postsynaptic spike, causing strong NMDA current right when the neuron is depolarized, triggers CAMK2 autophosphorylation and overwhelms the phosphatase pathway, yielding LTP. So it's a very fine balance between the phosphatase pathway and the CAMK2 pathway. And this calcium threshold model is supported by experiments varying extracellular calcium or buffering internal calcium. Reducing calcium tends to bias towards LTD since the peaks are blunted, whereas increasing calcium ion availability biases towards LTP. Now, NMDA receptor subunits tie in here as well, like I mentioned earlier. The NR2B rich receptors keep calcium flowing longer, which can actually help LTD because it promotes a more prolonged low level of calcium ions, unless the total crosses into LTP territory. And if NR2A rich calcium ions might come in a short burst that either makes LTP or if it's too weak, nothing. Now, interestingly, developmentally, it's observed that young synapses, which are NR2B rich, are very, very plastic, but also have lower LTP thresholds. As NR2A increases, the synapse becomes less inclined to potentiate from weak stimuli, which could mean a higher prosperity for LTD for modest activity. Now, this shows the importance of low sustained calcium spikes. Very, very important. So if a person is, for example, reactive or has very severe hyperacusis, it is extremely important that the stimulation, for example, is minimal, which will lead to a lower level of calcium spikes. So this means that it's extremely important that the somatosensory or the electrode stimulation and the auditory stimulation is not strong. It has to be the absolute minimum to achieve this prolonged calcium or low level of calcium influx. Now, besides NMDA receptors, the voltage-gated calcium channels can also contribute. Now, in some forms of LTD, particularly in spike pairing protocols with postsynaptic bursts, back-propagating action potentials or dendritic spikes activate L-type or T-type calcium channels to raise internal calcium ion levels. Now, these may be required in conjunction with NMDRs or certain forms of LTD. For example, in the cerebellum, paired pulse LTD relies on climbing fiber calcium ion signals. And as we all know, the cerebellum is very similar to the brainstem in terms of how the STDP works. But in cortical or hippocampal STDP, NMDA receptors are, of course, the main route. Now, one thing that I would like to mention that's pretty important is that subthreshold postsynaptic depolarizations can still trigger local calcium elevations that lead to LTD. It is not necessary for the postsynaptic neuron to fire a full action potential to induce plasticity. If the postsynaptic cell is held just below threshold and a presynaptic spike arrives slightly later, NMDA channels in the dendritic spine can open and let in calcium even without a spike. And this means that even stimuli that are not consciously perceived or did not cause the neuron to fire or the organism to notice, I guess, could be inducing LTD at synapses. For example, a very quiet tone that doesn't make an auditory neuron fire might still cause some glutamate release and NMDA activation in a partially depolarized neuron, nudging that synapse toward depression over repeated pairings. Now, this has implications for phenomena like masked inputs or adaptation. The brain can adjust synaptic weights due to timing of inputs that barely register. And some experiments have shown that brief subthreshold depolarizations in a postsynaptic cell paired with presynaptic spikes can induce long-term changes, sometimes termed subthreshold STDP. Essentially, as long as there is some calcium in the dendritic spine, even below the spike threshold, repeated coincident activity can accumulate to activate phosphatases. And this is actually very important with people who have hearing loss, because if you have hearing loss, this could mean that even though you do not perceive the auditory stimulation, there could still be calcium activity or low prolonged calcium activity that leads to LTD. So you don't have to crank the volume all the way up, for example, on the Susan Shore device. Now, once activated, phosphatases PP1, PP2A, PP2B act on multiple targets to express LTD. They dephosphorylate AMPA receptor, GLU-A1 subunits, at sites critical for channel conductance and interaction with scaffold proteins, causing AMPA receptors to be removed from the synapse or endocytosed. They also affect scaffolding proteins like AKAPs or stargazin, and can reduce the spine's sensitivity. Additionally, CREB, as we mentioned before, gets dephosphorylated, which may alter gene transcription, leading to long-term maintenance of the depressed state. PP1 and PP2A appear to act at slightly different timescales in LTD. PP1 is early, PP2A is later, possibly maintaining LTD after induction, which is very interesting. Now, a summary would be for LTD initiation via STDP, the timing sets up a scenario of moderate calcium ion influx post-spike preceding the presynaptic glutamate, and lack of strong depolarization, which preferentially triggers a cascade of phosphatases. And the combined action of calcineurin, PP1, and PP2A leads to synaptic weakening. If timing or receptor properties deviate such that calcium influx is too low, no plasticity occurs. If calcium ion influx is too high and too fast, then kinesis will dominate, or LTP. There is a Goldilocks calcium ion range for LTD. Because this process can occur even without overt spikes, neurons can undergo timing-dependent depression due to subliminal inputs, contributing to fine-tuning and also to maladaptive weakening, or in other words, sensory deprivation can induce LTD at unused synapses simply through lack of coincident firing, which is what I meant when I said that people with tinnitus or reactive tinnitus or hyperacusis get better in silence. And understanding these calcium-dependent pathways is very important for devising the interventions. For instance, drugs that slightly enhance calcineurin or inhibit CAMK2 could promote LTD in circuits where you want to reduce synaptic strength, like in, for example, if we're talking about memory, like in erasing a fear memory or dampening the tinnitus-related hyperactive or hypersynchronized synapses. And indeed, metaplasticity, or the plasticity of plasticity, I guess you could say, often involves adjustments of these calcium ion thresholds via the NR2A or NR2B ratio, or via neuromodulator-driven phosphorylation of NMDA receptors. Now, if any of you guys made it this far, congratulations. I expect most of you did not watch all of this, but I will be putting all of what I said today in kind of a paper format that can be read easily and referenced to later on, so wait for that. There is a lot of stuff that I wanted to say about fusiform cell synchrony and the sensory gating mechanisms in the thalamus, specifically the thalamic reticular nucleus. Also, I wanted to say some stuff about thalamic cortical dysarrhythmia, but we'll leave that to a later video because this one's already getting to be about an hour long, and yeah, I guess that's pretty much everything I wanted to say today about this. But basically, it is very important that spike timing is modulated for any bimodal or bisensory stimulation treatment device, because one timing will not work for everybody.